Before going to the main topic, I would like to explain the RBC structure and its adaptabilities. First, it contains hemoglobin. Hemoglobin transports oxygen from the lungs to all parts of the body. Second, it contains no nucleus or other internal organelles, which can carry more hemoglobin that leads to increased transport of oxygen. The third characteristic will be RBC as a circular biconcave shape. This will increase surface area to volume ratio and maximize the rate of gas diffusion. Lastly, the RBC is small and flexible. Thus, the RBC can squeeze through narrow capillaries. We can study the blood composition after centrifuge. The blood is composed of plasma and foam elements. Plasma is 55%, whereas foam elements is 45%. Plasma is composed of proteins and water, whereas foam elements is composed of RBC, white blood cell, and platelets. Now let us look into the principal functions of RBCs. Firstly, hemoglobin in the RBC transports oxygen from the lungs to the other tissue cells. Secondly, it picks up carbon dioxide from the tissues to the lungs. Thirdly, it contributes to the blood viscosity. And lastly, it is responsible for the acid-base buffer of the whole blood.
Hi, Assalamualaikum. My name is Syafierah and I will explain my part, erythropoiesis. Erythropoiesis is a process which human erythrocytes are produced. So basically, erythropoiesis is a process of red blood cells production. It is triggered by erythropoietin or EPO. It is a hormone produced by kidney when our body is in hypoxia state. Erythropoiesis takes place in the bone marrow usually, where the hematopoietic stem cells differentiate and eventually shed the nucleus to become reticulocytes. It is a process where hematocytoblast is transformed into a committed cell called proerythroblast. Okay, now let's take a look at the erythrocyte synthesis. Erythropoiesis can take place in different sites of our body throughout the life. So for fetus, in the first trimester, the production is in the yolk sac. The second trimester is in the liver and spleen. The third trimester is in the bone marrow of central and peripheral skeleton. While in adulthood, it can only occur in vertebral bodies, sternum, ribs, pelvic bone, sacrum, and proximal femur. In case of erythrocyte is inadequate, which means the body do not have enough red blood cell to transport oxygen to the whole cell in the body, this condition will trigger extramedullary hematopoiesis, which means formation of red blood cell outside of the bone marrow. Okay, in the slide you can see I put two diagrams which show the flow of process of erythrocyte synthesis. You can see the process start with the stem cell, which is the hematopoietic stem cells, HSC. HSC then differentiate into BFUE, CFUE, like in the flow, and then proceed to the proerythroblast until in the last step it will develop into a fully mature red blood cells, or we call it as erythrocytes. This process consists of three main phases. Each of the phases contain one highlighted thing that we will discuss later. So, okay, let's start. Okay, let's get started on how red blood cells being synthesized. Before we enter the phase 1 of the synthesis, hematopoietic stem cell first differentiate into more committed erythroid progenitors, which is the BFUE, burst-forming unit erythroid, from common myeloid progenitor cells. BFUE and then differentiate into colony-forming unit erythroid, CFUE. CFUE then convert into proerythroblast, as you can see in this diagram here. I put it down here. CFUE has receptor for EPO, the hormone kidney produce. EPO then later stimulate proerythroblast into erythroblast. So common characteristic about proerythroblast is it have high nucleus to cytoplasm ratio, which means the size of nucleus is much bigger compared to the cytoplasm. Okay, let's see the phase 1. One thing that been highlighted in the phase 1 is ribosome synthesis in early erythroblast. So there is differentiation of nucleated precursor from proerythroblast into basophilic, polychromatic, and orthochromatic erythroblast. We can refer at this flow down here. Basophilic turn into poly and then turn into orthochromatic erythroblast. So proerythroblast becoming basophilic erythroblast. Basophilic, it appear bluish because of it filled with mRNA as product of the transcription process of DNA. And then basophilic will becoming polychromatic erythroblast. Polychromatic, it appear more reddish compared to the basophilic because of the mRNA is translated into protein, which is hemoglobin, which then forms hemoglobin. Hemoglobin gives the red color to the polychromatic erythroblast. So, synthesis of ribosome in early pro-erythroblasts indicate the start of phase 1 of the process. Production of ribosome is very important for synthesis of other significant organelles needed by red blood cells, such as heme and globin, which then form into hemoglobin. Heme part is synthesized in a series of steps in the mitochondria and cytoplasm of erythroblast, while globin is also synthesized by ribosome in the cytoplasm of erythroblast. So this is the phase 1 about ribosome synthesis. Let's proceed to the phase 2. Okay, phase 2 is about hemoglobin accumulation in late erythroblast and normoblasts. As I said earlier, polychromatic it is more reddish compared to the basophilic. This is because the hemoglobin accumulation. So the characteristic here is hemoglobin accumulation, progressive decrease in cell size of the pro-erythroblast. As you can see here, the size is decreasing progressively from the stem cell until the polychromatic erythroblast. The size becomes smaller. This is because the cell lose some of its organelles. That is why the size is decreasing. And then the next characteristic is nuclear condensation. The nucleus become condensed. As you can see here, the size of nucleus becomes smaller. This is the phase 2, and then we proceed to the phase 3. In the phase 3, it is about ejection of nucleus from normoblasts and formation of reticulocyte. Normoblasts are also known as erythroblasts. Obviously, it comes from pro-erythroblasts. So polychromatic erythroblast will turn into orthochromatic erythroblasts. Orthochromatic erythroblasts will appear more reddish compared to the polychromatic erythroblasts because it contains more hemoglobin compared to the polychromatic, because as the process goes by, the production of hemoglobin is increasing, so yes, it will appear more red. Also, the nucleus of the orthochromatic erythroblasts will become more condensed as the cell body ready to kick out the nucleus from the soon-to-be blood cells. As you can see here in the picture of orthochromatic erythroblast or erythroblast, the size of nucleus is really condensed as it becomes so small. So as the nucleus is condensed enough, erythroblasts will inject the nucleus out of the cell along with some more organelles. So as soon as it loses the nucleus, erythroblasts will then change into reticulocyte, which is an immature red blood cells. Reticulocytes sometimes release into the peripheral circulation. Okay, please note that before this step, before reticulocyte, all the previous process is happening in the bone marrow. So sometimes reticulocyte is released into the peripheral circulation. So this is the phase 3 about the ejection of nucleus. Let's see how about the next step, the last step. Okay, the last step, reticulocyte will lose all the remaining organelles in it as it matures into erythrocyte, which is a fully mature red blood cell. So, as a mature erythrocyte, it is supposed to be enucleated because of the nuclear extrusion during the maturation process, the ejection of nucleus. And mature erythrocyte also supposed to be do not have any organelles remaining in it in order to maximize the transportation of gases. However, in case of any nucleated red blood cells found in sample of blood, it indicates that the release of incomplete development of red blood cells. So this may be a diagnosis for certain blood disorders such as thalassemia, severe anemia, and hematological malignancy. Also, yes, that it is. So the last step is the erythrocyte, which do not have any nucleus, do not have any organelles in it. So yes, that's it.
For growth factors of erythropoiesis, there are three major categories, which are general factors, maturation factors, and also extrinsic factors. For general factors, first we have hypoxia. Hypoxia is the body condition when we have low levels of oxygen. So hypoxia will stimulate the EPO secretion to regulate the erythropoiesis. Next, we have growth inducers, which are comprised of G-CSF, GM-CSF, interleukins, and EPO. For vitamins, we have vitamin B, vitamin C, D, and E. These vitamins are very essential for erythropoiesis because if we have deficiency of these vitamins, it can cause anemia. So erythropoietin or EPO. EPO is a hormone which is produced by kidney to stimulate the erythropoiesis in bone marrow. EPO gene can also be found in human chromosome 7. EPO helps to protect red blood cells from destruction once they are stimulated into the body. Low level of EPO indicates anemia and any chronic kidney diseases, while high level of EPO may indicate polycythemia, kidney tumor, and EPO abuse or blood doping. So blood doping is basically used to enhance athletic performance and endurance. So as for the maturation factors, we have vitamin B12, intrinsic factor of Castle, and also folic acid. So vitamin B12 is very important for DNA synthesis and also cell nutrition. It will form complex with intrinsic factor of Castle to so that they can be absorbed into our body. So any deficiency of vitamin B12 can lead to pernicious anemia. As for intrinsic factor of Castle, it is produced in gastric mucosa of parietal cells of gastric glands, and as I have said earlier, it will form complex together with vitamin B12. So deficiency of intrinsic factor of Castle or IF can cause severe gastritis, ulcer, and also gastrectomy. Lastly, for folic acid is very important for DNA synthesis and cell nutrition, and folic acid can prevent megaloblastic anemia. So for extrinsic factors, it comes from our diet intake. So we have vitamin C, protein, iron, copper, and others. So for vitamin C, it is very important because vitamin C helps to absorb iron and also plays a vital role in formation of hemoglobin. For protein, it consists of amino acids, which is the building block for globin synthesis. And as for iron and copper, iron is very important for heme synthesis, and copper is also needed for iron absorption, same as vitamin C. When there is any imbalance in blood oxygen level, stress erythropoiesis will be stimulated due to hypoxia. Hypoxia will stimulate the release of erythropoietin or EPO from kidney, and then EPO will stimulate the red bone marrow to produce more red blood cells. So our body will return to normal function when there is adequate oxygen level.
Welcome and hi, I'm Adira binti Mat Rofi. So I'm going to explain about detection of erythropoiesis related to disease. First lab test that can be done to detect the disease related to the erythropoiesis, which is red blood cells count. RBC count is the total number of red blood cells that are present in the sample of the blood. For normal RBC count for men, which is 4.7 to 6.1 million cells per microliter. However, for women, which is 4.2 to 5.4 million cells per microliter. As we all know, RBC counts can be used to diagnose blood-related disease such as iron deficiency anemia. As we all know, low RBC count due to bleeding, decreased RBC production, and increased RBC disruption. However, increased RBC count due to decreased plasma volume, increased red cell mass, and decreased hemoglobin in anemia. Next, we move to the second test, which is hemoglobin test. Hemoglobin test is to measure the concentration of hemoglobin in the blood. As we all know, normal concentration of hemoglobin in males, which is 8.7 to 11.2 millimoles per liter. However, for female, which is 7.4 to 9.9 millimoles per liter. If hemoglobin levels are abnormal, it is sign of having blood disorder. So for this slide, we can see the signs of low hemoglobin levels and high hemoglobin levels. If hemoglobin levels are low, it is signs of anemia, thalassemia, and iron deficiency. And for high hemoglobin levels, it is signs of lung disease, heart disease, and polycythemia vera. Third test, which is hematocrit. Hematocrit is to measure the ratio of the volume occupied by the red cells to the volume of the whole blood. This test is applied to screen the anemia and detect the polycythemia, while packed cell volume, which is PCV, is determined by centrifuging a capillary tube of whole blood and comparing the heads of the settled RBC to the heads of the column of the whole blood. Like we can see the normal range for PCV value and what happens if low PCV value and high PCV values. So normal PCV value for men, which is 38.3% to 48.6%, while for women, which is 35.5% to 44.9%. If low PCV value, it indicates that anemia, overhydration, and blood loss. However, for high PCV value, it indicates of polycythemia vera and dehydration. For the next test, which is erythrocyte sedimentation rate, which is ESR. ESR value is determined by measuring the distance from the surface meniscus to the top of erythrocyte sedimented in a tube for 1 hour. Reference range for 1 hour for adult men, which is 0 to 50 ml per hour, while for women, which is 0 to 20 ml per hour. For this slide, we can see that increased ESR value is found in tuberculosis and severe anemia, while decreased ESR value is found in polycythemia, allergic states, and sickle cell anemia. Okay, so for the last test, to detect the disease related to erythropoiesis, which is red cell index. Red cell index are sum of full blood counts tests. These RBC index are used to diagnose and classify of anemia and pre-screen tests for some disease such as thalassemia. First element in red cell index, which is mean corpuscular volume, which is MCV. MCV is the average size of RBC. Normal value for MCV, which is 87 plus minus 7 femtoliter. Normal-city anemia can occur when RBC count is decreased but size is normal. And normal-city anemia can be found in hemolytic anemia and aplastic anemia. So for second element in RBC index, which is mean corpuscular hemoglobin, which is MCH. It is the average hemoglobin content of RBC. Anemia with high MCH we call as macrocytic anemia, while low MCH can cause microcytic anemia. It is occur when the blood cells are too small. Next, we identify the mean corpuscular hemoglobin concentration, which is MCHC. MCHC, which is the average hemoglobin concentration in a given PCV. For normal value, MCHC, which is 33 plus minus 1.5 gram per deciliter. Low MCHC, it means that hemoglobin concentration per red blood cell is lower than normal. We call as hypochromic. while high MCHC means that hemoglobin concentration per red blood cells is elevated. Fourth element in red cell index that we can identify, which is red cell distribution weight. Red cell distribution weight, which is the degree of red cell size variations in a blood sample. Normal value, which is 11 to 14.5%. Normal red cell distribution weight, which indicates homogeneous population of cells, while red cell distribution weight over than 14.5%, it indicates that heterogeneous population of the cells.
My name is Muhamad Hazif from Sepian. I would like to present about a pathological condition or any related disease to erythropoiesis. First of all, anemia, which is a common blood disorder that occurs when the body has fewer red blood cells than normal. Lack of iron commonly causes this disorder. Iron is needed to produce hemoglobin that help red blood cells to carry oxygen to our body. There are many types of anemia, but we focus on the sickle cell anemia and aplastic anemia. For sickle cell anemia, it is caused by the mutation in hemoglobin beta gene that found on chromosome 11. It causes a sickle shape of red blood cell. Next is aplastic anemia. It is a rare but serious condition when bone marrow stops making enough red blood cells due to damaged stem cell of bone marrow. The symptom of anemia is dizziness, pale skin, and fatigue. Polycythemia is a condition where there is increasing in the number of red blood cells. The extra cell causes the blood to be thicker. In turn, it increases the risk of other health issues such as blood clots. The causes of this is by polycythemia vera, which is a rare, slow-growing blood cancer due to a mutation in gene. It causes the bone marrow to create excess precursor blood cells that develop and function abnormally. It will lead to the production of too much red blood cells. The symptom is unusual bleeding, itchy skin, and fatigue.